Hello, everyone, and welcome to today's live webinar, Understanding and Ameliorating Radiation-Induced Damage to Oligodendrocytes, presented by Dr. Alexandra Taraboletti. I'm Christie of Labyrinths, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labyrinths and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com/cell culture heroes. Now let's get started. Today's webinar is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them in the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, just use the Ask a Question box to let us know you're having a problem. If you have any trouble seeing or hearing the presentation, just let us know you're having a problem by using the Ask a Question box. This presentation is educational and thus offers continuing education credits. Click on the Continuing Education Credits tab and follow the process to obtain your credits. I now present today's speaker, Dr. Alexandra Taraboletti, Postdoctoral Fellow, Georgetown University. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Taraboletti, you may now begin your presentation.
Thanks, Christy, for that great introduction, and thank you so much for hosting me as the GIBCO Cell Culture Hero. Today, I hope to host a really accessible talk that covers the basics of white matter damage from radiation therapy with the title Understanding and Ameliorating Radiation-Induced Damage to Oligodendrocytes, and also go into how I use cell culture and metabolomics to better understand this issue. But before I dive into this topic, let me first tell you a little bit about myself. My bachelor's is actually in chemistry. I've kind of journeyed a far way. I got my bachelor's at University of Central Florida, and I started in all things in material science, where I did mechanochemistry, doping materials with catholuminescent properties. Tangentially, I also worked with crickets of all things, so if anyone ever needs to sex a cricket, I'm that person. I use these to look at temperature changes in respect to global warming. So again, very diverse background. But the chemistry and the biology led me to go into biochemistry. My PhD is in metabolomics and neurobiology. Technically, my degree is in chemistry from the University of Akron. But I looked at global metabolomics, and I investigated glial metabolism, which I'll kind of get into the glial part a little more here soon. But generally, I looked at neurodegenerative diseases, treatment of neurodegenerative diseases, and analyzing the metabolic products of some of these oligodendrocyte cells. Now I am a postdoctoral candidate. I have a postdoctoral fellowship at Georgetown University, and I study radiation metabolomics. One of my projects in particular is actually distinguishing metabolic biomarkers for radiation injury. This was looking at a pre-processing approach for ionizing radiation injury. So we were able to pick out biomarkers that allowed us to indicate injury and made a type of extraction that's useful for an event, a radiological event, to pick out injury markers. But today I am going to talk about the marriage of all of my fields of interest and knowledge, those again being the central nervous system damage, radiation injury, and then using metabolomics as well to kind of talk about all of these. From this overview, you should find the intersection of all three of those knowledge branches as we discuss, again, understanding and ameliorating the radiation damage that occurs in brain oligodendrocytes following cancer radiation therapy using the technique of metabolomics. Some of the learning objectives for this talk that I hope you will gain is just a general understanding of the current state of brain radiation treatment, patient outcomes, and their quality of life that follows therapy. I think this is important to always bring it back and think about patients. I also want to detail basic glial cell biology and how I use the MO313 cell line to model these immature and mature differentiated oligodendrocytes. So they're kind of the bread and butter of my work. And also understanding mass spectrometry-based metabolomics and how we can use it to monitor markers of radiation injury and oligodendrocyte metabolism in cell culture. So first I'm going to start with going into understanding the brain radiation treatment. So to talk about radiation damage, I first have to talk about cancer. Cancers of the CNS include cancers that affect the brain, the spinal cord, and also the cranial nerves. Today I'm going to focus mostly on talking about the brain. Primary central nervous system cancers can derive from different cell lineages. These include cancers like gliomas, astrocytic tumors, and oligodendroglial tumors, and also medulloblastomas. Tumors can also start in another part of the body and metastasize to the brain. These tend to be the most difficult to manage cancers and the most dangerous. They are also more common than the primary tumors that I previously mentioned. Treatment options for any type of CNS, whether it be a cancer that is metastasized or a primary tumor, include things like surgery. That's when possible to resect it from the brain. You can do a complete or partial resection of the tumor. Chemotherapy as well, so chemotherapeutic drugs, but these aren't used very often because it's difficult for many to cross the blood-brain barrier and get into the brain, excuse me. And then lastly is radiation, which is most commonly used. You can penetrate the brain from the exterior. You don't have to worry about the blood-brain barrier, and there's no complexities of surgery as well. Radiation treatment consists of the conventional therapy, which uses an external beam. It can be a beam of X-rays, gamma rays, or protons, and this depends on the type of cancer. It also actually depends on the facility, and in some cases it depends on what country you're in. Some countries only will allow certain types of ionizing radiation. This includes things like even carbon, which I believe is not currently being utilized in the United States right now. But generally, you use CT scanning to kind of get an idea of where to target the tumor, and then the ionizing beams are aimed at the tumor to kill the cancer cells and to shrink the tumor. This therapy is usually given over a period of several weeks, so it's fractionated, meaning multiple doses over time. And the actual area that receives radiation treatment may be large or small, meaning a small area of the brain or a large area of the brain. And that, again, depends on the type of cancer. Radiation treatment is usually limited to what we call partial brain irradiation when possible. But whole brain radiation is also common when presented, especially with metastatic cancer in the brain. So again, metastatic cancer, which has moved from another place, usually tends to form multiple areas of tumors instead of one singular tumor. And those are harder to singularly target. So a whole brain radiation, whole beam is used preferentially in this case. Whole brain treatments has widespread effects, as you might guess. Instead of targeting one single area, you're just irradiating the whole brain. So it causes more severe damage, and it also particularly impacts these stem cell populations in the brain, which is of real danger. So some of the injuries that have been documented and occur in stages that are from radiation therapy start with acute injury, and these can be things as simple as headache and drowsiness. And then you see kind of early delayed effects where demyelination has been documented. You see attention deficits and short-term memory loss. And then late documented injuries include vascular abnormalities, again, demyelination in the brain, loss of the brain cells or gliosis, and the most dangerous, cognitive impairment. So just general cognitive impairment seen over a long time. And this leads to poor quality of life in patients. So currently there is a limited focus on kind of ameliorating or stopping the damage caused by radiation therapy, which again leads to this poor patient quality of life when you have severe cognitive effects. These are just words I'm saying, but as a reminder, I've talked to patients that are involved with therapy. I've spoken with advocates and those who have survived, and a lot of the focus, of course, is put on survival of the cancer. But now a lot of these patients, now that they're surviving, they suffer cognitive impairment, which, again, hasn't really been focused on. So that's my goal of this study. Some direct examples of these injuries include the radiation-induced injury to children. So a large study was done by King et al. That looked at children who received radiation therapy for cancer treatment when they were young, of course, and into adulthood, these children unfortunately had declining IQs if they received radiation treatment versus if they did not receive this whole brain radiation treatment. Studies have also shown this cognitive impairment after whole brain irradiation in adults as well. So this Green, Schlosser et al. Also denotes that increasing after months of whole brain irradiation, you see almost 85% of the population having cognitive impairment. And again, this is after whole brain irradiation. One of the markers of this cognitive impairment that was previously mentioned and is tied to the memory problems and, again, cognitive impairment has to do with demyelination or loss of white matter. White matter is just a type of matter within the brain. You have gray matter regions and white matter regions. This demyelination is marked in animal models and also in humans as well. Here you can see from one of my studies, this is decreased staining in the corpus callosum. The stain stands for white matter. This is 7 days after 7-gray of X irradiation. The corpus callosum in particular, which I'm staining, is an area that's rich white matter area. And, oh, excuse me, went too far there. You can see, again, denoted in a mouse model, markers of white matter cells that are lost from 1 day to 15 months after radiation. You see both two markers, this O4 and MVP marker, which drastically decrease after radiation. And to confirm in humans as well, tissue from humans, these again are markers of white matter cells. So again, you see this demyelination that's persistent. And this marker seems to go along with the cognitive decline. So just giving the importance that the demyelination and white matter loss is connected to the cognitive decline that you see in patients. To get a grasp on radiation-induced damage in the brain, we focus on this white matter damage, and again, its connection to cognitive impairment. So we model the radiosensitive cells that actually make up the white matter. This is what our lab focuses on, are the cells of the white matter and their connection to this cognitive impairment. Which brings us into kind of our second learning objective, which is talking about glial biology, these cells of the brain, and how we use the MO313 cell line to model immature and mature differentiated oligodendrocyte cells. So the brain is not just made up of neuron cells, though they're very important. They're important for signal transduction and memory. There are other glial cells, we call them glial cells, that make up a bulk of brain tissue. And these include things like astrocytes. These are important for structure and metabolic support of neurons, and also the glial cell microglia. And these are actually the specialized immune cells of the brain. And lastly, and my favorite, of course, is the oligodendrocyte. These cells protect the neuronal axons. They help facilitate the signal that's transferred through neurons, so they act kind of like the insulation on a wire. And they also serve, they have a metabolic purpose, so they metabolically serve the neurons as well. So a lot of important roles. These cells, they wrap their cell membrane around neurons, around the axon. You can kind of see in this picture here, this spiral wrapping, to form what we call myelin. So just the wrapping of the membrane around the neuron is called myelin. And white matter regions in the brain, which we're interested in, are highly concentrated with myelinated neurons. So more generally saying, these areas are areas with more oligodendrocyte cells, which is why we're focused on the oligodendrocyte cells. They kind of are the cell of the white matter. So the death of oligodendrocyte cells is what causes demyelination. So what we were talking about before in the demyelination patterns, it causes that white matter loss and radiation injury and is connected to the cognitive impairment that we're worried about in patients. So our lab is interested in kind of examining these oligodendrocyte cells. Oligodendrocytes, as I noted, are also a metabolic support for neurons. They support a lot of the metabolic demands for neurons through production of things like pyruvate, this N-acetyl aspartate, or NAA, and other small molecule metabolites. The myelin sheath itself, which again is just the membrane of the oligodendrocyte, which wraps around the neuron, is formed highly of lipids, and it also has a very high energy cost to make all of those lipids and wrap them around the neuron. So these cells themselves are very vulnerable to energy depletion. Oligodendrocyte cells also start as a stem progenitor state, which is called the oligodendrocyte progenitor cell, or you'll see OPC. And they mature to the final version, which is the myelinating version, the version that actually wraps itself around the neuron. The OPCs, or stem cells, are more vulnerable to oxidative stress and damage from radiation, but the mature cells are harder to regenerate once lost. So kind of maintaining the balance of these populations is key to the healthy white matter, and also understanding how both of these populations act in radiation is also important. So to do this, we turn to a cell line, and the advantages of an oligodendrocyte cell line as opposed to using a primary culture. Again, it's a simple system. You have reproducibility when you have a cell line and very controlled conditions. And in particular, for when we're doing mass spec analysis, you can isolate individual metabolic features, which again is very key. We're very focused on how specifically the oligodendrocyte cell is reacting to radiation and not necessarily astrocytes or microglia or neurons. So those are all important, and the components of each of those and their metabolic interests are also important. We want to first start with isolating what is making the oligodendrocyte cell die. Looking at just a singular cell also has a less complex matrix, which is good for mass spectrometry. And we're using a cell line, you have the ability to collect high cell counts, which as opposed to primary cells, those are a bit harder to culture and to get going. For mass spectrometry, you need a lot of cells, and so the cell line approach in this case is very useful. So our lab uses and has developed the model of the MO313 cell line, which we use in DMEM with F12. It is a human oligodendrocyte cell line, and here's what it looks like. The cell line, one of the nice things about it is that you are able to look at both a immature state, so more like the OPC state, and the mature state of the cell. So it has this tunable advantage, and you differentiate it simply by using PMA added to the media. And here you can see the immature versus the differentiated state. They both produce markers of the oligodendrocyte, but you can see that their membranes kind of drastically change. You also see, if you look to the right, they're expressing this myelin basic protein, which is MBP. It's more highly expressed in the differentiated state, and that's associated with the myelin-forming sheath. For the first time, we've also used this cell line to model radiation injury. So here you can see the cell line in a sham state. This is receiving no radiation versus when undergoing 4-gray of X-ray. This is about 7 days post the X irradiation. The proliferative capacity decreases with increasing radiation, and you can see that these cells, they apoptose generally, they halt division, and they kind of flatten out. This is comparable to what primary cells do as well. So this model, for the first time, we've shown comparably modeled radiation injury in a cell line, just like how it is modeled in primary cells. So to recap, we've talked about radiation therapy and how it can cause long-term damage to white matter and cognitive decline. We looked how damaged white matter is composed of radiation-sensitive oligodendrocyte cells, and how the MO313 cell line can be used to model the radiation damage to oligodendrocyte cells. So with use of this MO313 model in hand, we can begin to explore the impact of radiation on oligodendrocyte function and its metabolism. Remember, these are metabolically active cells, so understanding their metabolism is very important. And also to model modes of protection. This is a great model to test out therapeutics. It's, again, very important to understand how the oligodendrocyte bioenergetics regulate normal brain function and how this dysfunction contributes to the end demyelination. So that brings us to kind of our last talking point, which is understanding mass spectrometry-based metabolomics and its use in this. Its use in monitoring the markers of radiation injuries and oligodendrocyte metabolism. On a cellular level, radiation causes this oxidative damage that starts an inflammatory cascade in cells, and that leads to cell death which then causes the demyelination or white matter loss, and potential long-term cognitive impairment. And again, oligodendrocytes are very susceptible to metabolic or oxidative changes, and this includes radiation. That seems to be why they are the most sensitive of the glial cells in the brain. So to monitor the oscillating metabolism of the oligodendrocyte cell in response to damage, we use this technique, metabolomics. And metabolomics is a technique that can monitor all small molecule metabolites, which are just small molecules. So think things like glucose or tryptophan or serotonin, all these neurotransmitters, any small molecule. And the difference of metabolomics and the importance of it is that the information is phenotypic, and this is as opposed to things like genomics, which is measuring at a genome level, or transcriptomics or proteomics. This is phenotypic information, and it also gives you kind of an instantaneous snapshot of the cell metabolism, which makes it very useful for temporal studies as well. So mass spectrometry metabolomics is utilized via two main approaches, which are targeted versus untargeted approaches. And again, it's useful for monitoring the injury that occurs in these oligodendrocyte cells. So in an untargeted technique, you take your tissue or cell in this case, and then you're going to detect all of the metabolites that are there. And then we do what's called statistical reduction and identification. So we identify all of these peaks we get into actual compounds. So we identify them as things like serotonin or tryptophan, et cetera. And then we have to do a validation step as well, just to validate. We check it against a standard. And in a targeted approach, instead of collecting everything, you're going to calibrate the machine to the metabolites of interest. So if you're only interested in neurotransmitters, or you're only interested in things in the tryptophan pathway, then you would do a targeted approach. And similarly, now the machine's just calibrated to the metabolites of interest. You detect those of interest, and you can quantify how much of that metabolite is present. So metabolomics kind of has endless applications. In this case, we usually are doing untargeted techniques to start with, just to look at what's happening in the cell. And then from there, we moved to the targeted approach, in which now we kind of have an idea of what we're interested in, and so we target and quantify those small molecules in the cell. So in some of our work, we've utilized metabolomics to analyze the capacity of drugs to ameliorate the inflammatory damage to oligodendrocytes. So again, it's important that we're using this cell line paired with metabolomics to be able to monitor these drugs' capacity to kind of stop inflammatory cascade. Generally, the idea of these projects is to induce oxidative stress and inflammation. This can be from radiation, of course, but we can also monitor other ways of oxidative stress. And as an example, one of the drugs that we're looking at is this dimethylfumarate, or DMS, compared to a vehicle. So doing a comparative analysis. And we use mass spectrometry again, this mass spectrometry-based metabolomics. And we're able to analyze all of the inflammatory molecules and all the changes in small molecules that occur, whether we give a protective drug versus vehicle. So in this example that I've shown, pre-treatment with DMS actually altered the TCA cycle, which is an important metabolic cycle in the oligodendrocytes, and it increased, you can see malate, fumarate, and flexonate. This is specifically with pre-treatment of the drug dimethylfumarate. You can see up to 72 hours of pretreatment gave you the most increase in this TCA cycle metabolite. And this is important because, as I keep stating, oligodendrocytes have high metabolic demands, and these TCA intermediates are crucial to its function. You can see kind of the positive impact of the drug using metabolomics on the cell line. You also see the protection through upregulation of glutathione. Again, if you just pretreat with the drug, we've shown that there's increased reduced glutathione levels. This is an antioxidant molecule, so it's helping to decrease the oxidation that could occur from radiation and kind of prepare the cell for oxidative damage. And we showed that when you pretreat the cells with DMS, as our metabolomics led us to believe, this increase in antioxidants also led to protection of cells when they're treated for up to 72 hours prior with the drug from oxidative damage. In this case, shown here, published is when they were treated with hydrogen peroxide, which is just another inflammation-inducing treatment. We then moved, of course, to look at radiation. Now that we kind of had an idea that using metabolomics in the cell line, we could see protection. And over time, there was an initial boost, again, in fumarate, succinate, and malate from the drug dimethyl fumarate when we used pretreatment, but it decreased generally after radiation energy. However, you can see that the levels of the antioxidants, glutathione, the reduced glutathione, actually stayed constant throughout up to 72 hours after radiation. This again kind of shows the power of metabolomics to monitor these temporal changes in small molecules and to oligodendrocyte metabolism in this response to radiation injury. And complemented by our metabolic data, you see that pretreatment of this DMF actually decreases cellular apoptosis following the exirradiation at 4 gray, so decreased cell death or loss of oligodendrocytes. So thus we kind of modeled a candidate drug, which is DMF, that can ameliorate the damage of these white matter oligodendrocytes, and we did this using mass spectrometry-based metabolomics paired with our MO313 cell line. So in summation, radiation therapy can cause long-term damage to white matter and cognitive decline, and damaged white matter is composed of these radiation-sensitive oligodendrocyte cells. The MO313 cell line, which I showed you, can be used as a good model for radiation damage to oligodendrocyte cells and is well paired with mass spectrometry-based metabolomics to help model products of radiation injury in oligodendrocytes. I've also shown you kind of a case study in which we showed that dimethyl fumarate can offer protection to oligodendrocytes through metabolic alteration. So I'm so thankful to be, again, chosen as a GIBCO Cell Culture Hero, and I'm thankful to everyone that I've worked with at Georgetown University, in my Tumor Biology program at the University of Akron, where I started my PhD, and for all the funding that has supported me as well. And with that, thanks for listening, and I want to open it up to any questions. Thanks.
Thank you, Dr. Taraboletti, for your informative presentation. We will now start the live Q&A portion of our webinar. Now, if you have a question you'd like to ask, Please do so now. Just click on the Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. We have some great questions coming in. Dr. Taraboletti, what exactly is the mechanism of action for DMF?
Oh, great question. Thanks, Christie. So DMF is, let me start, actually already a known drug that's being used. It's a neuroprotectant. I'm actually using it as a repurposing drug. So it's FDA approved in use of multiple sclerosis. Its mechanism of action is actually to target NRF2, which then leads to the upregulation of glutathione, which is one of the reasons we're looking at glutathione production.
Thank you. Now our next question. How could these therapeutics be used for patients receiving radiation therapy?
Okay, so as an actual application for the therapeutics, because most of our work was looking at a protective effect that occurred when treated prior to radiation, this would be a type of drug that would be given to patients who, again, are going to be going in for radiation treatment and therapy about 72 hours prior to the actual radiation that they would receive, and then hopefully give a protective effect.
Now, are there any other fields in this research that have application to outside of cancer therapy?
Yeah, so one that I would, any occupational field in which people are receiving radiation and potentially could receive radiation to the brain. These are also, it's a great area to study for protection in those events. One thing that I'm passionate about, because I'm actually a big space nerd, and my parents worked at NASA when I was young, so one area that's being explored also for these types of protectants is for astronauts that are going to go to Mars. They're going to need protection from radiation as well.
We've got some great questions coming in. Let's go with this next one. Why would you use targeted versus untargeted mass spectrometry in your study?
Okay, so I kind of touched on this as far as like what untargeted and targeted is. Untargeted being you're looking at exploration of all metabolites, but it's not, it's semi-quantitative, it's not truly quantitative in that respect. So if you want to actually quantitate the metabolites that you're looking at, or how much, in a simple sense, how much are there, you'd want to use a targeted experiment. It's also just more sensitive. So kind of once you know what you're looking for, in this case, I kind of understood that TCA molecules are being changing in my MO215 cells using an untargeted first metabolomics experiment, and then I shifted over to targeted now that I know what I'm looking for, and I can actually see how much is there.
Thank you, Dr. Taraboletti. Now, what metabolites do you see in this inflammatory cascade?
In the inflammatory cascade, so in this particular cell line, and again, this is because it's an NRF2 target, this I'm looking at glutathione production, but much of the work in my lab has been directed at this inflammatory cascade that happens with radiation in general, and not necessarily in relation to using DMS as a protectant. And so in that case, you would see some of the more standard inflammatory molecules in the work that you're doing. I suggest that you check out the Fornace lab and see kind of what they're working on.
Thanks, Dr. Taraboletti. Now we're almost out of time. We'll have to wrap with this question. Would DMS be protective with grade 3 to 4 brain tumors at 60 gray IR?
That's a good question. This is far kind of in the future as far as patients that we'd be looking at. I have looked at this in respect to glioblastoma, which is a very aggressive brain tumor. And there have been actually some patient studies that have looked at DMS not as a neuroprotectant, but actually against, as an anti-cancer agent, I should say. And in that case, it didn't work as an anti-cancer agent, but they did see some protection. I don't know about at the 6-week ray on my end in my research, just because we haven't tested at that amount. Ideally, right now, we're mimicking in mice, which isn't an exact model, the amount of radiation they would receive for a glioblastoma treatment. But I haven't tested it at that high of radiation.
Thank you, Dr. Taraboletti. Now, do you have any final comments for our audience?
I just want to give a thanks again. Thanks to you, Christie. Thanks to Gitto in general for letting me do this talk. It's been a really great experience. And of course, if anyone has further questions, you want to reach out to me, feel free to look at my website at ataraboletti.com, or you can find me all over Twitter as well, in the science Twitterverse. So, thanks.
Thank you again. Now, we'd like to thank our audience for joining us today and for their interesting questions. Again, just a reminder that the questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. Thank you again to Dr. Alexandra Taraboletti for her time today and her important research. We'd like to thank LabRoots and also our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's time for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now, and we hope to see you again soon. Have a great day.